So the continuing with my video, so this is what I talked about in my previous video, that is the structure of contractile proteins, right? If you recall, just now we have finished with the structure of our contractile proteins. This is our thick filament, which is having the chain and these globular heads. So as I told you that this is the our tail portion which is visible to us, and these globular heads, they are exposed. These globular heads, they are exposed. So these are our globular heads because they have to bind with these actin filaments. Now this is the structure of our actin. Actin has the G proteins as the monomer. Now as you can see their arrangement, let me look at it. So here these circles, they are coiled, they are overhanging with each other, and this fiber thread like structure which is moving around them, that is called tropomyosin. Then there is the sites, these are the pockets on which globular head has to attach. These are the pockets on which globular head has to bind. But यहाँ पे कौन बैठा है resting stage में? ट्रोपोनिन. यहाँ पे कौन सी प्रोटीन आके ब्लॉक करके बैठी है इसको? ट्रोपोनिन. ट्रोपोनिन एंड ट्रोपोमाइसिन टुगेदर दे ब्लॉक द एक्टिव साइट्स of the for the myosin on the actin filament. Is it clear? This much is. Now let's move on to our next very important contraction mechanism, which is explained with the help of sliding filament theory. That is given by three scientists, that is A.F. Huxley and J. Hansen. They describe these sliding filament, right? And the why I'm saying that we need to discuss this sliding filament theory, because ultimately in your syllabus we have to talk about the mechanism of muscle contraction, and without sliding filament theory you can never describe the mechanism of muscle contraction. It is always best explained with this theory, right? Actually, which states that what is the main criteria, what is the main concept behind this? It says that contraction of muscle fibers, these you can add in your notes also. Contraction of muscle fibers, it take place by sliding of thin filaments over थिक फिलामेंट्स. मीन्स हमारा जो कंक्लूजन निकलेगा वो ये निकलना चाहिए कि हमारे जो ये ग्रीन कलर के पिक बनाए हैं मैंने, थिन फिलामेंट्स, दीज आर योर एक्टिन फिलामेंट्स, दीज आर गोइंग टू स्लाइड ओवर आवर myosin filaments, right? So how is this thing going to start? But first you have to always draw this structure. As you can see, there is the Z line, and between the distance between two Z line, it is known as sarcomere, and the portion where only myosin is present, it is known as H zone. Then the M line, which is made up of myomycin, protein myomycin, it is known as the M line. Now actually what is happening is that there is a cross bridge which is forming between actin and myosin. There is a cross bridge which is forming between actin and myosin. That is what we are going to discuss now. So when muscles, how does this start? See, any muscle contraction, it starts when it receives a signal. And you people are very confident that our skeletal muscles, they are under control of CNS. It means at the neuromuscular junction there will be release of neurotransmitter, right? It means who is sending this signal? This signal is sent by CNS via motor neuron. This signal, it starts with CNS via motor neuron. Now a motor neuron that connected to this motor unit, which is called your neuromuscular junction, right? This is the it is connected with, there will be a junction, there will be a gap between the neurons and the gap, that is the signal. Who receives the signal? That is the sarcolemma. So sarcolemma, they will receive the signal, and a neural signal, it reaches at this junction, और इस junction पे पहुँचते ही it releases chemical, and that chemical is neurotransmitter, and most of the time it is acetylcholine. अब ये क्या करेगा? It generates, what it generates? It generates action potential impulses in the sarcolemma. और sarcolemma से कौन लेके जा रहा है impulses? T-tubules. Now this is spread through the muscle. These impulses spread through muscles by T-tubules. Are you getting my point? That what is happening, that a neuromuscular junction, it receives the stimulus in the form of neurotransmitter, which is acetylcholine. So it generates action potential in sarcolemma. This spread the impulses throughout the muscle, and it caused that now your T-tubules get activated. So these T-tubules, they start releasing calcium ions, right? और कहाँ पे release करेंगी? They release calcium ions into sarcoplasm, right? Now, when there is increase in calcium ions because calcium ions release होना शुरू हो गए, so with increase in calcium ions, now this troponin start binding with the calcium. अब इसका मतलब कि जो ट्रोपोनिन यहाँ पे बैठी थी, ये यहाँ से निकल के किसके साथ बाइंड कर गई? कैल्शियम के साथ. इसका मतलब इसने ये हमारी पॉकेट्स को खाली कर दिया, राइट? ये हमारी पॉकेट्स खाली हो गई. नाउ पॉकेट्स खाली होना मतलब there is a cross bridge is formed between now actin and myosin site, क्योंकि जो भी हमारी active sites थी for myosin, now they are free, right? They are free or exposed. So now the globular head, it will bind with your actin, right, on the site. So basically, as you can see, this is our myosin, और its head is exposed, but यहाँ पे actin present है, but it cannot bind with this actin क्योंकि यहाँ पे troponin और tropomyosin ने इसको block कर रखा है, right? As soon as calcium is released, अब calcium released हो गया, so troponin के लिए क्या है कि troponin जैसे ही calcium release हुआ, troponin की बहुत भाई bonding है calcium के साथ. ये बोली I am going with my best friend. Okay, so now it will leave the actin, boli tata bye bye. Now I will bind with my best friend, which is the calcium. So troponin bind with calcium. Myosin is really happy because उसके लिए यहाँ पे जगह खाली हो गई है. So it forms this globular head will set block in this. ये पूरा यहाँ पे ऐसे कवर हो जाएगा, ये इस पॉकेट को कवर कर लेगा, दिस कैन कवर दिस पॉकेट. सो देयर इज दिस actin myosin cross bridge is formed. Clear? So in this way a cross bridge is formed. Now what is happening? For this movement 1 ATP is required. राइट 1 ATP इज़ रिक्वायर्ड. सो 1 ATP जब टूटेगा तो हमारा क्या बनेगा? एक पार्ट से तो जो एनर्जी रिलीज होगी एक्टिन मायोसिन, ये हमारा एक्टिन मायोसिन ब्रिज बन गया, राइट? एक्टिन मायोसिन ब्रिज इज़ फॉर्म्ड. सेकेंड पार्ट of energy, second part of energy is used by myosin head to give a power stroke to actin. Basically what is happening that ये यहाँ पे एक्ट किया है, जैसे ही वो एक धक्का सा देगा तो दिस मायोसिन आगे आ जाएगा और दीज विल शिफ्ट. सो बेसिकली स्लाइड हो रहे हैं. सो मायोसिन का जो पॉकेट था यहाँ से यहाँ शिफ्ट हो गया, यहाँ वाला यहाँ शिफ्ट हो गया. सो देयर इज़ अ शिफ्ट, देयर इज़ कॉल्ड पावर. जैसे मैंने किसी को धक्का दिया, बेसिकली पुल्ड कर दिया. मैंने क्या किया, मायोसिन ने क्या किया, बेसिकली मायोसिन पुल्ड these actins, they are pulled inwards by the power stroke of मायोसिन. ये अंदर वाली साइड में एक्टिन फिलामेंट्स हमारे पुल्ड हो जाएंगे. राइट, अगेन वन मोर टाइम इन द फ्लो चार्ट फॉर्म अभी हम देखेंगे. यू विल बी मोर क्लियर कि व्हाट इज हैपनिंग. जस्ट लुक एट दिस. Actin has a sites, active sites which are blocked. Who is blocking them? Troponin and tropomyosin. This is the globular head which has to form a cross bridge. This is the myosin, it is the globular head. Now this is the condition when your muscles, they are relaxed, and this is the Z line which is bisecting the actin. Now just look here. Just look here. This is the when stimulus is received. Stimulus receive होने का मतलब है calcium will be released. Calcium release, then cross bridge will be formed. There will be cross bridge formed, and now the ATP is released. ATP टूटेगा. So once energy is finished, अब energy तो finish हो गई, cross bridge बन गया है already. Now head, जो head है, it detach. अब ये क्या करेगा, अब ये था हमारा, let me make it with the purple only. This was my myosin and this was the head which was attached to the active site. And ये जो मायोसिन था, अब ये जब जैसे ही हेड डिटैच हुआ एक्टिव साइट से, ये इनवर्ड पुल करेगा आपका एक्टिन. आपका एक्टिन जो है इस डायरेक्शन में इनर साइड की तरफ जाएगा. राइट, द मोमेंट योर हेड, हेड किसका हेड, ग्लोबुलर हेड ऑफ मायोसिन, इट डिटैच फ्रॉम द एक्टिव साइट, इट गेव अ पुश, इट गेव अ स्ट्रोक, धक्का देता है उसको, और उस धक्के की वजह से दीज एक्टिन फिलामेंट्स दे स्लाइड ओवर द मायोसिन. राइट, सो बेसिकली व्हाट इज हैपनिंग, सो आपका जब ये स्लाइड करेगा, now you people tell me when it slide, so length of sarcomere, ये थी हमारी, ये हमारा sarcomere था. right, now make it with red only. This was the sarcomere and these were our actin filaments and this was the मायोसिन. अब मायोसिन जब अपना हेड डिटैच करेगा तो ये जो एक्टिन है वो इनर साइड की तरफ मूव करेंगे, एंड जैसे ही ये इस साइड मूव करेंगे देयर विल बी द लेंथ ऑफ दी सार्कोमियर ऑल्सो डिक्रीजेज, एंड लेंथ ऑफ एच बैंड, एज यू कैन सी मायोसिन का पोर्शन कम हो गया, लेंथ ऑफ एच बैंड ऑल्सो डिक्रीजेज. ए बैंड को कोई फर्क नहीं है, ए बैंड रिमेन्स द same. So here basically what is happening, that actin filaments, the two more points you need to understand here, that actin filaments, they pulls towards the center. Right, they are pulled towards the center. Now Z line attached to these actins, इसके साथ कौन attached है, Z line to these actins, they are also pulled inwards. So sarcomere will be shortened or not? Yes, sarcomere length decreases and this is called our contraction. Right, it is called contraction. अब now what has happened? Energy use हो गई. Now myosin release ADP and PI, inorganic phosphate, and now it again ये बोल रहा है कि अब मुझे जाने दो. It is again going back to relaxed state. So क्रॉस ब्रिज टूटेगा कि नहीं? यस, द मोमेंट हैड डिटैच, क्रॉस ब्रिज इज ब्रोकन एंड अ न्यू एटीपी हैज़ टू हाइड्रोलाइज. एक और न्यू एटीपी को टूटना पड़ेगा टू फॉर्म क्रॉस bridge. So in this way this happens one by one, with one after the another, that first the cross bridge forms with the head and actin, then it globular head detach, then again there is the with the energy cross bridge is formed. So this cycle, this goes like a cycle. So this cycle, it is helping in the sliding of actin filaments, right? This is called the sliding of actin filaments. Now once basically the process, ये कब तक continue रहता है? Till your calcium, till the calcium ions are pumped back to sarcoplasmic reticulum. Sorry, बीच में चला गया है ये. So but you can write down that this process continues till calcium ions they go back to sarcoplasmic reticulum, राइट. सो जैसे ही सार्कोप्लाज्मिक रेटिकुलम में कैल्शियम वापस जाएंगे, नाउ अगेन एक्टिन बेचारा अकेला रह गया. सो दीज एक्टिन फिलामेंट्स अगेन, नाउ एक्टिन अकेला नहीं रह गया, आपकी ट्रोपोनिन, कैल्शियम गया वापस, यहाँ पे बाइंडिंग थी ट्रोपोनिन की, ट्रोपोनिन वापस आके एक्टिन फिलामेंट्स अगेन दे आर मास्क्ड और ब्लॉक्ड बाय troponin, and this is called relaxation. This is called relaxation. So contraction में क्या हो रहा है? Contraction में हमारा क्रॉस ब्रिज बन रहा है, and relaxation में हमारी active sites they are getting blocked, right? So this is the main process of our muscle contraction with the help of sliding filament theory. The moment your actin filaments actually slide, then this length of sarcomere decreases, length of H band decreases, but A band remains same. I hope that you are able to get my point, that what do I want to say here, right? So this was about our muscle contraction with the sliding filament theory. Go through this video one or more times, you will be able to get your easy that information, right? Easily able to understand your concept. Page number 308 and 307, if you will read after my lecture, everything will be correct, everything will be properly will go in your mind. So please read the page number 307 and 308 of your NCERT.